Well, good afternoon, everyone. I'm Eric Green. I'm director of the National Human Genome Research Institute at the National Institutes of Health. And I'd like to welcome you to the 28th lecture of the NIH Genomics and Health Disparities lecture series. By way of reminder, the first lecture was held in May of 2015, and we've carried on ever since. And the series aims to highlight the opportunities of genomics research to address health disparities. The series is co-sponsored by the National Heart, Lung, and Blood Institute, the National Institute of Diabetes and Digestive and Kidney Diseases, the National Institute of Minority Health and Health Disparities, the National Human Genome Research Institute, and the Office of Minority Health and Health Equity at the Food and Drug Administration. Now, speakers have been chosen by the series co-sponsors to present their research and their thoughts on the ability of genomics to improve health of all populations, and often the challenges in making sure these improvements are accessible and applicable to all populations. Speakers in the series have approached this problem from different areas of research, including basic science, population genetics, translational research, clinical research, and social science research. Well, we're really pleased to have my good friend and colleague, Dr. Iftikhar J, joining us this afternoon, and it's my pleasure to introduce him to all of you. Iftikhar is a clinician-investigator or a physician scientist in the Department of Cardiovascular Medicine at the Mayo Clinic in Rochester, Minnesota. He heads the Atherosclerosis and Lipid Genomics Laboratory, chairs the Cardiovascular Genomics Task Force, and directs the early atherosclerosis and familial hypercholesterolemia clinics at the Mayo Clinic in Rochester. His research is focused on the genetic basis of atherosclerosis and lipid disorders, as well as implementation of genomic medicine. For the latter in particular, he's been funded by the NIH since 2003, but in particular relevant for NHGRI, he's a principal investigator in our electronic medical records and genomics, or eMERGE program, and also our Polygenic RIsk Methods in diverse populations, or PRIMED network. All of this is funded from NHGRI. Iftikhar also is really a valued colleague of the Institute. He's actually a member of our advisory council, in other words, the National Advisory Council for Human Genome Research. So he's going to give his presentation, after which there will be a facilitated discussion. And I would just ask you to submit your questions at any time via the Q&A box. But in the meantime, please join me in welcoming today's speaker. And Iftikhar, why don't you take it from here?
Thank you very much, Dr. Green. I'm really honored to have this opportunity to speak on how we might work towards equity in genomic medicine. As a disclaimer, I'm a clinician investigator, as Eric said, and my focus is on the genetic epidemiology of coronary heart disease and lipid disorders. But I have also been heavily involved in genomic medicine implementation, primarily in my role as a PI in the NHGRI's eMERGE network. So I don't claim to be an expert in equity or social. I'm not a social scientist, but I hope to convey to you my experiences in observing existing disparities in genomic medicine, and then how we might move towards equity in all of these areas. So in part one of my talk, I'll describe some of these disparities that I have encountered while attempting to implement genomic medicine. And in part two, I'll describe work towards making genomic medicine more equitable. My focus in that latter part will be on polygenic risk scores, which are of particular interest to me. So in the first part, I'll talk about disparities in variant interpretation, population screening, laboratory reference ranges, inclusivity in clinical trials, and also polygenic risk scores. So at the outset, I want to acknowledge my colleagues in Mountain Park Health System, Phoenix, Arizona, which is a federally qualified health system, and Mayo and my lab have had a collaboration with colleagues at Mountain Park for nearly a decade. And as part of that, we have really learned a lot together about the challenges in implementing genomic medicine in low resource settings. First, I want to describe disparities in variant interpretation. Of course, traditionally, genomic medicine has involved testing for Mendelian disorders when there's clinical suspicion, and that typically includes sequencing of a panel of genes. And there are concerns that when we interpret these findings, that we may not be interpreting them correctly. And much of this was spurred by this article in the New England Journal in 2016, which showed that variants that were labeled as pathogenic for hypertrophic cardiomyopathy for individuals of African ancestry were actually wrongly labeled. So because the prevalence of these variants in this population was really quite high enough that they would probably be classified as benign. So individuals of non-European ancestry more often receive false positive tests, but they may also receive more often ambiguous tests such as variants of uncertain significance and even false negative, because sometimes there are not enough data for these populations in our data repositories. And more often, their variants may be reclassified over time. So the ClinGen has created a diversity and ancestry work group to address this issue. And their recommendations broadly are to include diverse populations, patients in genomic studies, and importantly, standardize race, ethnicity, and ancestry measures, and to be mindful of information disparities across populations when curating or interpreting variants. An example would be that absence of a variant in NOMAD for a particular population may simply be because there's not enough sequencing data for that population in that repository. Another disparity lies in population screening. While increasingly we are doing more genomic screening in large cohorts, which often includes a diverse population, which is great. But one of the things we encountered is that once you screen individuals in low resource settings, one should also have a plan in place for a follow-up and medical care of these individuals from underserved communities. So this is a story of a woman in her 40s from a federally qualified health center who, as part of a genomic screening research study, was found to have a likely pathogenic variant in the fibrillin gene, indicating that she possibly had Marfan's. And when we went into her history, she indeed did have a history suggestive of Marfan's with ectopia lentis, bilateral hip joint arthritis requiring replacement. Unfortunately, when this finding was conveyed, the patient was not able to afford CT imaging of the chest. She did indeed mention her dramatic family history, including a father who died of a ruptured aorta, a sister who had a replacement of her aorta secondary to rupture, another brother most likely also died of a ruptured aorta. And despite this, the diagnosis had not been established in the family. She herself was able to get many of her family members this information, but there were a lot of difficulties in actually trying to establish her aortic dimension. And finally, since she could not afford a CT and ultrasound, which I presume an echocardiogram of the chest was done, and thankfully her aortic dimension was within normal limits, but obviously she will need close follow-up. Stories like this in the eMERGE phase 3 prompted the NHGRI in phase 4 to include medical cost coverage for federally qualified health center participants. Another disparity is the difference in reference ranges for circulating biomarkers. And these reference ranges are typically derived from small samples of predominantly individuals of European ancestry. But as I'll try to show here is that they are probably not appropriate for individuals of diverse populations. In this slide, I've summarized work that took us nearly 4 years to complete. We measured 47 candidate biomarkers in pathways that are relevant to cardiovascular disease in a cohort of nearly 1,324 African Americans and 1,237 European ancestry individuals. Now, shown here are beta regression coefficients from multivariate linear regression analyses. And when these bars are to the right, that means the levels are higher in individuals of African ancestry. And when they're to the left, it means they're lower in the population. And you can see that for many pathways, individuals of African ancestry have higher levels, whereas for some of these markers, they are in the opposite direction. Now, we tried to adjust out as much as we could these differences. So, for example, we adjusted for age, sex, obesity, renal function, medication use, prevalent disease, hypertension, diabetes, smoking, lipids, alcohol, physical activity, education. Yet these differences were statistically significant and quite strong in many of these biomarkers. So although we did not, it's quite possible that unmeasured confounders may still be playing a role. It's likely that genetic factors influence these differences. And I'll just zoom in on a couple of these markers from the previous slide. First is lipoprotein A, and the other is D-dimer. Both of these are measured often in clinical settings, and therefore reference ranges that are not appropriate will have clinical downstream implications. So lipoprotein A is a biomarker for cardiovascular risk. In fact, it's been recommended that this should be universally measured in adults. And you can see on the left side that individuals of African ancestry, shown in black, the levels are nearly threefold higher. It's a really dramatic difference in both men and women. On the right, you see D-dimer, which is a marker of fibrin breakdown and often indicates active clot formation. And you can see that although less dramatic, these differences are still significant statistically. And the lipoprotein A levels are measured for cardiovascular risk, so you can imagine the consequences if the reference ranges are not appropriate. Whereas D-dimer is used often in the emergency room when a patient is suspected of pulmonary embolism. So there may be potentially inappropriate testing in individuals of African ancestry if there are no appropriate ranges relevant to that particular population. This actually extends to many lab measures, and it's well known that white cell counts are lower in individuals of African ancestry, and often this leads to unnecessary bone marrow biopsies. Hemoglobin and other red cell traits may also differ in individuals of African ancestry. And then differences in LDL, HDL, and triglycerides are quite significant in this population compared to other populations. And physiologic measures also might be different. For example, lung function test measures may differ among populations. And extending this to clinically used tests of physiology and imaging, the first being the ankle brachial index, which is used to diagnose peripheral artery disease. And almost 2 decades ago, we showed that there were significant differences between individuals of African ancestry and European ancestry for this measure, both in men and women. So the top dotted line is white men, and this solid line here is African-American men, and you can see a very profound difference here. And this was also true in women, although less dramatic. These differences actually occurred even at younger ages when atherosclerosis is unlikely to have developed in the legs. And so most likely these may be related to arterial physiology and wave reflection, and motivates the establishment of reference ranges that are different from what is currently in place. On the right side is another example of differences in imaging parameters. Here we see a CT scan for coronary calcification. These are data from the multi-ethnic study of atherosclerosis, showing that African Americans were 22% less likely than European ancestry individuals to have detectable coronary artery calcium. So these are important caveats and can lead to disparities in not only genomic, but regular general medicine. The next disparity is in clinical trial inclusion. And again, I'm going to refer to clinical trials here that are aimed at reducing lipoprotein A levels. So far, we have not had a specific therapy for lipoprotein A, which, as I mentioned, is an established risk factor for atherosclerosis. But genomic guided therapies have been developed. On the left is an antisense for apolipoprotein A, and on the right is a small interfering RNA against the same apoprotein. And these are both phase 2 studies. They're dose-ranging studies. The sample sizes are relatively small, about 300 in both studies. On the left is the study that was published about 2 years ago. On the right is a study that was published yesterday in the New England Journal and was just discussed at the HA, where I actually happened to be today. So the good news is that these therapies are very effective, and you have 80 to 100% reduction in lipoprotein A levels at the highest doses. The bad news is the representation of diverse populations. So you can see in the bottom tables that the percent of African Americans is very low in both studies. Now, we at Mayo will likely be involved in some of the phase 3. The next step will be phase 3 studies. And in my initial conversations with the clinical trial group, I have pointed out this disparity and encouraged them to try to be more inclusive in the phase 3 studies. And this is really important for this particular therapy, because it may be that the effect of these might differ in between certainly European ancestry and African ancestry individuals. And this is based on a Mendelian randomization study that we performed in nearly 1 million individuals using eMERGE Million Veteran Program and UK Biobank participants. And so we collected genetic variants that affect lipoprotein A or increase lipoprotein A levels. And we then estimated what was the association between a 1 standard deviation increase in genetically predicted lipoprotein A levels and the risk of incident coronary heart disease. And as you can see in the top part of this chart, the risk is quite significant in individuals of European ancestry, at about 28% higher for each standard deviation increase in genetically predicted lipoprotein A levels. But in African American individuals, this risk is lower and is quite modest. In fact, in this particular analysis, didn't really reach statistical significance. And this was true for ischemic stroke, but for these two manifestations of ASCVD, there was a more robust association in both populations. Finally, I'm going to describe disparities in PRS, and this has been a topic of great interest over the last several years. And astonishingly, in around 2009, about 96% of GEOS cohorts were of European ancestry. This improved to a decade later to about 22% of the GEOS cohorts being non-European, but unfortunately, only less than 5% of these cohorts are of African ancestry groups. So a major disparity, which unfortunately translates into lower performance of polygenic risk scores for many traits in individuals of African ancestry. And this is a study done by Dr. Dikilitash in my laboratory using the eMERGE cohort. And what he noted was in this cohort, which had about 45,000 European ancestry individuals, about 7,500 individuals of African ancestry, and about 2,500 individuals of Latino or Hispanic ethnicity, These are the numbers, ages, percent of women. The number of incident events is shown here, much less, as you can see in the latter two groups, and the mean follow-up, which was around 9 to 11 years. Now, for each standard deviation increase in the polygenic risk score, the risk in individuals of European ancestry was a 50% increment. But in contrast, in individuals of African ancestry, this was nearly half, only about 27% increase in risk with each standard deviation increase in the polygenic risk score. And this was a statistically significant difference. This is the p-value for the interaction. And this is just shown visually here, is the very great gap in the performance in the polygenic risk score for heart attack in these two population groups. So I've conveyed to you more, I think, a personal experience of these disparities that I have encountered while trying to implement genomic medicine. So I'll now move to the next part where I'll describe what is being done to reduce these disparities and move towards equity. Of course, if one considers these in the larger picture, we are facing very substantial challenges in terms of poverty and structural racism. But I think it will take a global effort and all of us individually making efforts to move towards equity. And I think in particular, proud of the NHGRI and the genomic community of the wonderful initiatives and efforts that have been implemented over the last decade or so to reduce disparities. Much of this part of my talk will focus on polygenic risk scores, but I think similar paradigms would be applicable to other domains as well. So why polygenic risk scores? Well, they have become an intensely active area of research with a major focus on their use for estimating disease risk, because they capture orthogonal and incremental predictive information with the potential to improve risk prediction for common diseases. And more importantly, they can be integrated with existing risk frameworks that might include monogenic information, family history, or clinical variables to generate comprehensive risk scores. In the last 5 or 6 years, there have also been major advances in polygenic risk scores, in part because GEOS cohorts have increased in size, and that has led to discovery of more genetic susceptibility variants. And the other major development has been what we now call the polygenic or omnigenic model of complex disease, where previously we were just taking genome-wide hits and composing polygenic risk scores. But for many diseases, particularly cardiometabolic diseases, it's clear that one can dip below this p-value threshold. And in fact, p-value thresholds as low as 0.1 or even 0.5 are being used. Essentially, this is what's known as a pruning and thresholding approach, where you dip down into the p-value thresholds and then throw out the variants that are in linkage disequilibrium to come to a score, which may include thousands of variants. And these scores have been shown to be much more robust, certainly for cardiometabolic disorders, than scores that are restricted. In other words, just to include the genome-wide significant variants. Another development has been the use of Bayesian methods, where a prior causal proportion is assumed, and different proportions are tested till one ends up with a score that performs the best. And so this has really motivated now the desire to implement polygenic risk scores in the clinical setting. To address the disparity in polygenic risk score among population groups, the NHGRI has formed the PRIME Consortium, as Eric alluded to earlier, And the main goals of this consortium are to develop methods and improve the applicability of polygenic risk scores across diverse populations, optimize the integration of large-scale, harmonize genomic and phenotypic data sets so that collaborations can occur and meta-analyses can be performed. This consortium includes 7 sites in the US, a coordinating center, and then partnerships with NHGRI and NCI. And Anvil, which is a cloud platform created by the NHGRI, will be the locus of collaborative analyses and tool development. And this map just shows that this consortium is reaching across the globe to create polygenic risk scores that are applicable to diverse populations. So I'll just, in the next 3 slides, describe 3 approaches that we are taking, we and others are taking, to make polygenic risk scores more robust. And one approach is to use functional annotation. As you know, when we have GEOS hits, only about 10% are in coding regions. So the 90% are intergenic and in regulatory regions. And some are assumed to be in regulate gene expression. And so we have now increasingly available annotation resources that are listed here, primarily ENCODE and GTEx. These resources can help us annotate some of these GWAS variants and perhaps increase the predictive accuracy of polygenic risk scores. In our laboratory's case, we are interested in polygenic risk score for CHD, and this is work that's ongoing in our laboratory at Mayo Clinic. The next approach is to capture a broader genetic liability. We are approaching this in 2 ways. Firstly, we are asking the question whether for coronary heart disease, in addition to the PRS that is specific for coronary heart disease, can we improve predictive value by including polygenic risk scores for the risk factors for coronary heart disease? In other words, blood pressure, lipids, diabetes, and BMI. And the rationale is that when you measure blood pressure and put it in a risk prediction equation, is just one measure single time cross-sectionally, whereas a polygenic risk score for that same trait might give an individual's lifetime exposure to that particular risk factor. The other approach we are taking is to look at correlated traits, and for coronary heart disease, those traits would include ischemic stroke, abdominal aortic aneurysm, and peripheral artery disease. These are all manifestations of atherosclerosis. And we are testing the hypothesis that by including risk scores for these traits, we would boost the predictive ability for coronary heart disease. And as you can see in this grid here, there are mild to moderately strong genetic correlations between coronary heart disease, abdominal aortic aneurysm, ischemic stroke, and peripheral artery disease. Lastly, we are very much interested in evolutionary signals. And as we all know, humans originated in Africa 200 to 300,000 years ago and spent most of that time in Africa. Before about 70 to 100,000 years ago, small groups went out of Africa and then populated the globe in different environments. And as a result of this, the need to adapt to different environments, and also as a consequence of genetic drift, we do see significant allele frequency differences in variants for among these population continental groups. Several years ago, we looked at about 16,000 common SNPs in 416 genes that were in etiologic pathways of coronary heart disease. And we looked at a measure of FST, which is shown here. This is just a toy example. In A, you have the red variant, which is only present in one population. And so as a result, when we compare the FST between these two, it's pretty significantly increased. In the second circle, B, the red variant is present in both populations, a little bit more in one, but not enough to be deemed statistically significant. Remember, this has to be done in the background of what allele frequency differences there are. in other neutral areas of the genome. And so we did this in about 416 genes in etiologic pathways of coronary heart disease, and noticed that the FST for SNPs in these genes was significantly higher when we compared Uruban versus Ceph samples and Uruban versus East Asian samples. These differences were not as significant in comparison between Ceph and East Asian populations. And as a result, after correcting for multiple testing, we identified 6 genes where the FST was significantly different. And these signals of selection might be useful in annotation of genetic variants and perhaps improve the performance of polygenic RIsk scores. This is another area that is worth exploring and something we are actively involved in. But no matter what functional annotation or other informatic approaches we apply, in the end, the bottleneck really is the number of GEOS cohorts for a particular population. And this is where we are lacking. And for individuals of African ancestry, unfortunately, these training cohorts are small in size, and therefore the polygenic RIsk scores are not as robustly associated with traits. So I would say that a very high priority for us in the genomics community is to increase the number of individuals of African ancestry, both in the continent of Africa and elsewhere, to genotype and create large cohorts, which are actually linked either to electronic health record or some survey that collects phenotypes of importance in these areas. In fact, at the last ASCG meeting in the presidential symposium, there was a whole session devoted to African genomes. And I think Dr. Collins made the pitch for a moonshot for African genomics, which I think is clearly the way to go. And taking the example of PCSK9, which is a drug that has really transformed cardiovascular practice, which was initially the variant was found in individuals of African ancestry. Perhaps pharmaceuticals companies might be interested in investing so that we make new discoveries that will benefit not only individuals in Africa, but elsewhere in the world. In East Asia, there are biobanks, the Biobank Japan and China Kaduri. There's also a biobank of Latin American population that's underway. And in the Middle East, finally, the Qatar and Emirates biobanks are also in the process of recruiting. Perhaps there's some lack in South Asian representation, but I'm told that the PROMISE study has collected decent numbers. And of course, in the US, we have the All of Us Million Veterans Program at TopMed. In all this excitement about genomics, we cannot forget the environment. And many of the common complex disorders that afflict mankind are really, to some degree, related to the modern environment. So as a species, we were hunter-gatherers, and our genomes perhaps were not well equipped to the modern ages of unlimited availability of food and sedentary lifestyle. So these ancestral alleles are, in fact, maladaptive to this modern environment and explain our predisposition to diseases such as diabetes, coronary heart disease, and obesity. And of course, unfortunately, you have to layer on top of that additional stressors of poverty and structural racism that play a significant role in susceptibility to disease in many areas of the world, including areas in this country. So increasingly, there is awareness that social determinants of health play a very important role in disease predisposition. And they include not only measures like sleep quality, exercise, but social factors like income, transportation, access to nutritious food, housing, education, air pollution, smoking, access to bike trails and running paths. And these act on the genetic profile we have, and also there are interactions with the environment that eventually lead to disease. But we have to recognize and acknowledge the profound role of environment in leading to disease susceptibility. And that's why we need to measure environment very well if we are to make progress in genomic medicine and disease prediction. And we have started with geocodes or zip codes. That's a low-hanging fruit. But there's excitement that wearables are strategically placed, even a watch or a sleep sensor or others that are shown in this cartoon here. These wearables can generate a lot of data that could be transmitted via mobile devices into the electronic health record. I believe an experiment of this nature will be conducted in a subset of all of us patients. And this is, I think, important if we want to make significant advances in being able to better predict disease susceptibility. The next area where we need to work on is admixed individuals and admixture. So we discussed polygenic risk scores for populations, but in reality, all of us on this planet are admixed to varying degrees, some more so than the others. In fact, the two major non-European populations in the United States are admixed: the African-American population group and the Hispanic Latino population group. This is, I think, a vast area in itself in terms of nomenclature and the implications. And I should note that the National Academy of Sciences recently had a symposium on this and will shortly publish those findings. But the point I want to make here is that we can look at global ancestry using reference panels and sets of genetic variants. And we did that in self-reported, we actually identified Latinos by self-report, but also by genetic analyses, and defined individuals who had more than 10% American Indian, American Alaskan Native genes as being genetically admixed admixed Americans. And we can see that there were individuals who did not report self-report as Latinos, but they indeed, from the global admixture ancestry analysis, could be classified as such. And then there were, in this section here that I'm showing with the pointer, those who self-reported as Hispanic Latino, but did not quite make the grade when we used 10% as a threshold. So this is just to point out the many nuances and intricacies of ethnicity, race, and ancestry. But one of the medical implications of admixture is, well, there are two. One is something we can leverage, which is perhaps better gene mapping and identifying disease susceptibility loci. But the other is a challenge of creating polygenic risk scores for admixed individuals who may have varying degrees of admixture of the parental populations. So when we do local ancestry, this is an example from eMERGE. This is an individual who is by self-report Latino. And you can see here that the areas in red, as we walk across the chromosome, these areas in red are of Native American ancestry. And there are areas in green here that are of African ancestry. So whereas global ancestry is not very helpful for admixture mapping, local ancestry might indeed be. And this is something we are testing in eMERGE, where we are doing local ancestry in about 5,000 individuals who are self-reported Latino. And you can see here that the amount of Native American admixture varies across the chromosomes. And at certain places, it's actually very high, more than 4 standard deviations from the mean. And we are exploring this closely to see if there are any phenotypic consequences related to these areas of increased local ancestry for Native American origin. Finally, I'll talk about implementation, and this is, of course, what the eMERGE network is doing currently in its phase 4 is implementing polygenic risk scores. The network consists of 10 academic sites in the US, a coordinating center, and then genotyping and sequencing centers. And there are 10 traits for which polygenic risk scores are being calculated, and then these are returned to 25,000 individuals, 5,000 of whom are children, and 50% of these participants are expected to be of non-European ancestry. The goal here is to really create comprehensive risk profiles, realizing that a polygenic risk score is just but one of the factors that lead to disease predisposition. And we are also doing monogenic testing for tier 1 conditions, namely breast and ovarian cancer, familial hypercholesterolemia, and Lynch syndrome. And then we have a polygenic test for 10 conditions. And we also get patient-reported data for lifestyle and other factors that are often difficult to get from the electronic health record. And then we have clinical variables from the electronic health record. And all of this input is then collected in a portal called R4, where computation algorithms then derive risk profiles called genome-informed risk assessment for each individual. For those individuals that are at high risk for one of the 10 traits, the study staff returns these results in person, and in the remainder, these are notified by letter and are also placed in the electronic health record. Our immediate goal is to look at near-term outcomes, most importantly, whether new therapies are initiated or new diagnoses are made, new screening tests are done, and also whether patients change their behaviors, health behaviors. And I'll just show an example for coronary heart disease, which is one of the 10 traits. So individuals will be classified as a high risk if they have a monogenic predisposition, namely familial hypercholesterolemia, or if their polygenic risk score is greater than 95th percentile. If they don't have these criteria but do have a family history, they are notified by letter, but otherwise they will be seen in person by a genetic counselor or by a study coordinator. And the outcomes for coronary heart disease are listed here. New tests or referrals, such as lipid profile, CT scan for coronary calcium, referral to a cardiologist, new risk reclassification or diagnosis, new drugs are initiated, or whether they make any lifestyle changes or share with family, or develop any anxiety or decisional regret. Eventually, of course, we will try to answer the important question of whether acting on this information actually reduces adverse outcomes. So in the final 2 slides, I'll summarize how we are moving towards equity in genomic medicine. I think for variant annotation, we need to keep sequencing more individuals of diverse populations, but at the same time, we want to standardize the intake of race, ethnicity, ancestry by clinical laboratories. We should continue to do more population screening, particularly those that include diverse populations, but ensure that appropriate follow-up and care is available when this is done in low research settings. I highlighted the importance of laboratory reference ranges, which were derived from one particular population group and perhaps are not applicable to other populations. We need to establish these range in diverse populations. We need to increase inclusivity in clinical trials. I'm at the AHA today, and when I open my smartphone app for the conference app, this is the picture I see. This is a particular company advertising a particular drug. But in reality, when we look at the participation of, say, African Americans in these phase 3 trials, it is quite low. And finally, we need to make polygenic risk scores more equitable. And this is, I think, summarized on this slide. We need to improve performance using novel methods. that utilize increasing knowledge about gene regulation. In particular, importance, I think, is the need to increase GEOS cohorts of African origin. We need to develop methods that we can make PRSs applicable to admixed individuals, perhaps by methods that include local ancestry and polygenic scores. We need to incorporate social determinants of health in risk prediction frameworks. And then finally, we need outcomes data so that eventually the clinical utility is established and payers can cover polygenic risk score in clinical practice. So I'm going to end here and acknowledge the many individuals that contributed to this work. In particular, I want to express my gratitude to the many participants of these studies and also to the NHGRI for funding most of this work. I think that the problems we face are daunting, particularly those of poverty and social structural racism. But I'm really inspired by the genomics community in that it's leading the way to address these issues in whatever ways and means it can. And I'm proud to be of this part of this community and proud to be, contributing to these efforts. And so I want to thank you for listening, and I'll be happy to take any questions. Thank you.
Thank you, Dr. Kullo, for that outstanding presentation. I'm Nishadi Rajapaksa. I'm a program director in the Health Equity and Global Health branch at the Center for Translation Research and Implementation Science at the National Heart, Lung, and Blood Institute. And I will be moderating the Q&A session. So again, I would like to remind the audience to please submit your questions via Q&A on the bottom of the page. So with that, I think maybe I'll start with a couple of questions. So Dr. Kullo, you had alluded to the understanding of social and behavioral environments of patients. In addition to genomic information can greatly inform clinical decision-making, right? So the social determinants of health, whether it's income, financial resource strain, education, health literacy, et cetera, are really important to provide that contextual context for understanding a patient's clinical phenotype. And then oftentimes, actually, the SDOH has even more direct impact on their health than genetic risk. So could you talk a little bit more about the integration of SDOH in the diagnosis of genetic disorders, in particular even through the addition of PRS in improving health and life expectancy for all?
Sure. Thank you for that question. I think that one of the positive developments has been that as we use EHRs almost universally in this country, that now the importance of these determinants of health has been acknowledged. And actually, whenever a patient has an appointment, there's typically a questionnaire about these social determinants of health. So, better late than never. I think we will hopefully accumulate, these data sets where these structural determinants have been collected in an organized, structured fashion. And I think that will really provide a lot of valuable insights into their contribution to disease predisposition. And I anticipate we'll be, surprised by the extent to which they play a role, while acting, on the background of our genetic makeup. So I believe that this is particularly relevant, of course, for common diseases, but one can make the argument that all genetic diseases are probabilistic. Even Mendelian disorders or monogenic disorders, there's an element of an environment. And so they may be relevant to even monogenic disorders, but certainly for common disorders, as well as oligogenic disorders, they might play a particular role. And I think easily what comes to mind is cancer, cardiovascular disease. I think these determinants of health we'll realize play a very substantial role in what may have been previously attributed to the construct of race or ancestry. We will realize that much of that was actually confounding by social determinants of health.
Right. And I think in particular, even your example about the woman with Marfan syndrome, I'm sure there were probably difficulties, access to healthcare in that instance that may have played a role in the fact that she was, never diagnosed, even with having particular gene variant that causes Marfan syndrome. Okay, so let's go on to a couple other questions. What do you consider the biggest challenges to obtaining equity in access to the health benefits of genomic medicine in the current environment?
I think the biggest challenges are, I would say, if we step back, then the challenges I alluded to are, things like inequity that is social in nature and is, again, poverty and structural racism. I think if we step back, those are huge challenges for the human race, not only just us in this country. But if we then go closer, I think in terms of genomics, assuming that we work towards addressing those major issues, I think it would be generating trust among underserved communities, indigenous communities, low-income, middle-income countries, to make them more engaged in genomic research so that they can benefit from precision medicine. Because I think that their engagement is critical. And once they are engaged, then having systems in place that will, recruit them, engage with them, and not just, do helicopter research where, you come, you take samples and are never seen again, but truly engage with the communities to address their needs, which may actually be different from the genomic assessment. But I think with proper engagement, one can make the case that that's also important. So I think generating trust and having in place many of the mechanisms that I described that will, if we don't correct them or put a check on them, will perpetuate or even widen these gaps, I think is important. And I see that there's a very great awareness, as I said, among the genomics community. And I see a lot of work and effort being put in by the leadership at NHGRI, but across the country and in other parts of the world, that we have suddenly realized that we may have gone a bit too far in one direction, and we need to kind of correct course and make sure that we, all boats rise with the tide. I don't really blame anybody. I think that's natural progression of science, and you work with what you have, but now we realize that we need to maybe just course correct a bit.
Yeah, no, that's true. Actually, at NHGRI, we really invested more recently in a platform to increase community engagement as a result of actually the COVID-19 pandemic. And so now there are a number of different initiatives under the community engagement platform, including climate change and health, something I'm leading. But of course, there are others with primary care, etc. So there's a huge interest within NIH for the community engagement platform. Okay, so I think the last couple of minutes I wanted to touch on. So in 2016, Pope Joy and Fulton's paper, and this was actually an update, I think, from Bustamante's original paper, that talked about there was a 300% increase in the proportion of GWAS participants in non-European descent, but still it only constitute about 19% of GWAS non-European individuals. So has there been much progress in improving these numbers in the past 5 years or so? And I know that these could have major implications for screening as well as other data gaps in the training sets as well.
I think we are making progress. I think I can point to all of us that data is going to come on board. In fact, it is already on board for a proportion of the participants. I think the Million Veterans Program is quite diverse. And Emerge, I think in its current phase, is also recruiting a substantial number of minorities. So I think we are making progress. It's not to the rate that we would like, but is definitely in the right direction. At the last ASAG meeting, I think there was clearly a great enthusiasm for the African Genomics Initiative. And I think that's really critical, not only for people of Africa, but I think the entire world, because we all originated in Africa, and there's so much diversity and genetic diversity in that population that we are going to make so many discoveries that will benefit all of humanity. So I think that slow, but in the right direction, and hopefully we'll see a really rapid upstroke with these initiatives that are about to be launched, hopefully soon.
Yeah, and so I have one final question for you, Dr. Kullo. What is your advice to trainees and students who are interested in pursuing their career in genomic medicine, just in general?
Absolutely. So I would really want to encourage students, particularly of diverse populations, minorities. The NHGRI has a lot of programs available at all levels that one can avail of. But generally, I think this is an incredibly exciting time to be in genomics. There are so many opportunities for people in genomics to improve diagnosis, improve disease prediction, and develop new therapies. We are seeing that in these antisense agents in CRISPR. And I think that this is an incredibly exciting time to be in genomics. And I can't think of a more exciting field to be in. So I really encourage students to think of actively training and participating in genomics research, and particularly those from diverse populations. I think they can really make a lot of help the whole field move forward.
Yes. So thank you again, Dr. Kullo, for a fantastic lecture and discussion. So on behalf of the Genomics and Health Disparities lecture series and co-sponsors, please join me in thanking Dr. Kullo for sharing his work and perspectives today. And then thank you for those in attendance for joining us and for your participation in this robust conversation. So the next presentation in this lecture series is scheduled for February 2023, and I invite you all back. Enjoy the rest of your afternoon. Thank you very much.
Thank you very much.